Hey, Miss Mandy, can I ask you a question real quick?
Yes.
Sorry, before class starts, I don't want to forget. But so as far as the lecture test goes and you taking our highest grade, the highest grade is out of 100 points. So for instance, like if we get a
No, Sydney.
oh yeah,
We have first two exam, it's 100 points, but
right,
third and fourth exams are 70 points, right?
yeah,
Yeah.
yeah,
So in that case, what I'm going to do, like out of 70, what's your percentage? I, I,
right, so,
I will make the
yeah,
percentage, and then I will
put
go by
that
the
to 100.
percentage. Mhm.
Okay, so like if you got a 60 out of 70, that'd be a 90. So you'd put a 90 out of 100.
Yep.
Okay, I see. Okay, perfect. I was just curious, and I know some other students were curious too after I asked them. So thank you. Just wondering.
Okay,
Thanks.
so we have to wait a little bit more. People are still joining. Let me share the PowerPoint there. Okay, let me stop video. Okay, I think it's 6:30. Let's start. Okay, so we are going to work on chapter 15. So this chapter talks about microbial mechanisms of pathogenicity. Means, what is virulence factor, how pathogenic the bacteria are, how are we going to measure the pathogenicity, means which one is, a really serious pathogen, which one is not, then what are the signs, symptoms, so all those things we are going to work on. So the disease, any condition in which the normal structure and function of body is impaired or damaged. Okay, so that is called disease. So any deviation from a condition of good health and well-being. Okay. So basically normal structure and normal function is impaired. So when it is a broken leg, okay, is it a disease? No. But over here, it is a deviation from a normal condition, right? But still, it is not a disease, because that broken leg, that's an accident. Okay, so that's not a disease. Now, infection, successful colonization of microorganisms in a host's body. So basically, when the microorganisms, they are successful to make colony inside our body, then we can tell that actually they made infection inside our body, or I have infection of certain bacteria in my body. So when they are our pathogen, bacteria or any microorganism, they are inside and they make us sick, the first criteria is they have to make colony. If they cannot make colony, that means our immune system got them. Okay. Once they can make colony, then only they can give us disease. Otherwise, they cannot. And to make colony, they have to go inside our body. Okay, they have to have one entry point through which they can go inside. Like for the coronavirus, every time we are talking about that. Okay, so drink hot water, because you know, if they are inside your throat and if they go through the digestive tract, then they cannot survive. Right? Why? Because if they reach at the stomach, it's highly acidic, and that environment they cannot tolerate. So they only be successful if they, instead of the esophagus, if they move through the trachea, then only they can you know, give us disease. They can make a colony inside our lungs, and then they can give us that disease. So
Dr. Nandi, so it is true what they say. I've seen several posts on Facebook, and they've sent me things over WhatsApp, that if you drink like hot water, like whatever you can stand and not burn, that you can destroy the coronavirus that is in your throat with the heat.
If you do gargling, you know, if you do gargle, and if you drink hot waters, then what happened? That can dissolve the fat of the virus, and yeah. So
okay, thank you.
So so basically, to give us sick, they have to make colony inside our body. So infectious disease condition caused by the presence or growth of infectious microorganisms or parasites. So when all those microorganisms, they make colony inside our body, that means they they give us that infection. And the disease we get from any type of infection, so means from the colonization of any microorganism, that is the infectious disease. Okay. So non-infectious disease. No, so before that, when we talk about the infection, so any colony made by microorganism, means made by any pathogen, gives us infection. So who are the pathogen? Any microorganism that can cause disease, who are capable of making colony inside our body. They are our pathogen. Non-infectious disease, genetic disorder, disease for the environmental factors, immunodeficiency disease. So basically, if we talk about the disease, so there are two different types of disease. One is infectious disease. That actually come from the microorganism. Means if they can make colony, if they can make infection inside our body, then we get that infectious disease. Other group of disease that are non-infectious. Non-infectious disease could be, let's say, it's from heredity. Hereditary disease, like you can say sickle cell anemia. Sickle cell anemia, you can say Huntington disease. All are hereditary disease because you get it from your parents, or basically you actually are going to inherit those diseases. So that's one type of non-infectious disease. Then from let's say environmental factors. Okay, so from environment we get, let's say, pollen. So because of the rise of pollen, we are sick. We get cold, cough, fever, watery eyes. So that's basically non-infectious disease. Okay, we really do not have any pathogen inside our body, and that's because of the environmental factor. Then let's say immune response. Okay, non-appropriate immune response. Okay, so in that case, example is allergy. That's actually, we get that allergic reaction because it's inappropriate immune response. Our body cannot differentiate between the foreign cell and our own cell, and that's why we get allergy. So immune response actually, you can tell, where is the pen and where is the color? Immune response, it could be like acquired. If it is acquired, then you can get that HIV. Okay, because you have that disease, and that is why your immune response is it's not appropriate. Okay, now another thing can happen. So I talked about heredity, environment, immune. I can talk about, let's say, malnutrition, or you can say lack of nutrition. That could be another type. Lack of nutrition. Okay, lack of nutrition, let's say, because of vitamin C, you are going to get that scurvy. So we are sick. It's not our regular normal body condition, but the reason is not any microbes, not any pathogen. It's because of the lack of nutrition. It's deficiency, nutritional or deficiency. So these are all disease, okay? But in that case, they are type of non-infectious disease. Sign, objective and measurable evidence of disease, as noted by an observer. So the person who is sick, he or she can feel, plus who is observing, any nurse, mother, parent, anybody can measure that, like vital signs like temperature, fever, heart rate, breathing rate, rash. As a nurse, you can take the blood report. You can, if you want to see the, if you want to look for the, let's say, RBC count. Okay, so yep, you can do that from the blood report. So all these are objective and measurable. So they are the signs. Symptoms, they're subjective evidence of disease as experienced by the patient. Only patient can feel, like chills, pain, any type of ache, body ache, headache, nausea, itching, fatigue. So you cannot measure that how much headache the patient has. You cannot. Okay, you can ask that. Okay, so if I give you a scale, number scale, like from 1 to 10, what is the, which one you think that how strong it is? Nowadays we are measuring all these, we are trying to measure actually all those symptoms, but the symptoms are actually subjective evidence. Syndrome, that's actually collection of signs and symptoms, often associated with a particular disease, like over there, it's a collection of signs and symptoms, like, let's say, septic shock, toxic, what do you call, toxic shock syndrome, I think. Toxic shock syndrome. In that case, what happened, like fever, then rash, skin peeling, what else is there for toxic shock symptom, low blood pressure, so all low BP. So all these together, fewer signs, fewer symptoms, all these together, when all these signs and symptoms are together, we easily can tell that, okay, so it is toxic shock syndrome. Like for coronavirus, like if you have high temperature, high fever, then if you have dry cough, okay, if you have the fatigue, so all those together are you can tell that these are the syndrome, flu syndrome. Like we know that all these signs and symptoms must be there. Asymptomatic or subclinical means disease do not show any noticeable signs or symptoms, like herpes simplex virus, that it's called herpes simplex virus. We have two different types. We have HSV1 and HSV2. We have shingles that we get from the pox virus. So this disease for long period of time, you can harbor these viruses or microbes, any type of microbes, but for long period of time there won't be any signs and symptoms. Herpes virus 1, suddenly that virus actually comes out, and infection around the mouth, lips. You can see the cold sores, all those, and it's very, very contagious. It moves through the kissing, sharing utensils, same glass if you use. HSV2, that's the genital herpes. Okay, in that case, what happened, you can see the sores around the genitalia or rectum. So but in both the cases, herpes virus, for long period of time, it does not show any signs and symptoms. Okay, the person who is carrying the virus, he or she does not even know that he is carrying that virus. So that's the asymptomatic or subclinical diseases. Nomenclature, like what happened, many syndromes are named using the nomenclature. Using the nomenclature means based on let's say signs, symptoms, or maybe the location of the disease. Okay, that's how we give the name. So the suffix emia means presence of an infectious agent. Okay, bacteremia means presence of bacteria, fungemia, presence of fungus, but the septicemia means presence of an infectious agent in the bloodstream. Okay, so the difference between septicemia and bacteremia. So bacteremia, presence of bacteria inside your body. We really do not know where the bacteria is. Septicemia, the presence of, like if it is bacteria, means it's inside the blood stream. So when any microbes, they can invade our body all the way up to the blood, when they can move through the blood, so that is the septicemia, and that septicemia can come from any microbes. The suffix itis meaning inflammation, like pharyngitis, inflammation of the pharynx. So over here, the word pharyngitis indicates two things, like the type of the disease, what happened, it is a type of inflammation, and the place of the disease. Okay, where did it take place? It's inside the pharynx. Endocarditis, inflammation of the heart chambers. Suffix lysis means destruction, hemolysis, so destruction of red blood cells. So like you all know, like that blood agar plate, we looked for alpha, beta, gamma hemolysis. So in that case, that bacteria can destruct or degrade the red blood cells. Suffix pathy means disease, like neuropathy. Neuro means nerve. So in that case, the disease actually affecting the nerve. Suffix oma meaning tumor. Lymphoma means cancer of the lymphatic cells. Okay, so this is the place where it took place, and the symptoms, what happened actually. Now the classification of disease, okay? Communicable disease. Communicable disease capable of being spread from person to person through direct or indirect contacts. So direct contacts or indirect contacts. Direct contacts like the person sitting behind you or besides you. That person is sneezing, coughing, and you can pick up that air drops from the when you are actually breathing. So that's the direct contact. Indirect contacts, when you are going to get the disease from fomites. Okay, so basically from the doorknobs, from the bed sheets. Now the coronavirus, you can get it from the shopping bag, shopping cart. Okay, in that case, that shopping cart or that poly bag, all those works as fomites. In that case, they actually carry the pathogen. Okay, so that's the if you get it from there, that means it's from indirect contact. It's like now for the coronavirus, if you pick up your letter from the letter box, you can get it. So that's the indirect contacts. Now the communicable disease, you can get it directly from a person, okay, or you can get it indirectly from the fomites. So all are communicable disease. Okay, so directly from person, like as I told, like flu virus, like the person is sneezing, coughing, and you can pick up the air drop directly from the air, or from the indirect contact, like from your desk where you are sitting in class. So basically, the person who was sitting over there at the same spot before your class, like class before yours, and she was sneezing, coughing, and after that class, when you sit on the same spot from the desk, you can get that virus or bacteria. So that's the indirect contacts. Contagious disease easily spread from person to person. Okay, like measles. In case of measles, virus can quickly move by the droplets. Okay, so contagious disease, they are also capable of spreading from person to person, but in this case, it's very easy to spread. So what's the difference between then communicable disease and contagious disease? All contagious disease are the communicable disease. Okay, they are capable of being spread from person to person. So all contagious disease, they are a type of communicable disease. But not all the communicable diseases are contagious. Okay, like let's say gonorrhea. Gonorrhea. Now gonorrhea, it is a communicable disease. You can get it through really intimate, from very intimate contact, you can get it. But it has to be very intimate contact. Then only you can get it. So it is a communicable disease, but it's not contagious. It's not easily spreadable. It has few conditions that needs to be fulfilled. Versus the measles, that's contagious, because you can get it very quickly from the air, from the environment, from the fomites. You cannot get the gonorrhea from the fomites, from the air. You cannot get it. So to get that on, it is communicable, but it's not contagious. Okay, so that's the difference between communicable and contagious. I hope you got the difference. Iatrogenic disease. Iatrogenic disease occur after a medical procedure. Okay, medical procedure, like let's say it could be surgery, it could be wound treating. You have a cut, so you had to go to get the stitches. Okay, and after that you can get infection. That can happen. Catheterization, if the person catheterization, so from the catheter, people can get the infection, and catheterization occurs because of the surgery or any type of procedure. Okay, so that one way. Then another way to get that iatrogenic disease means, let's say you can say the side effects of drug. So you had that surgery, and that's why you are taking that drug, and it's a side effect of drug. Or you can say the complications from the surgery. It can happen. Okay, iatrogenic disease.
Dr. Nandi, are these the same as opportunistic infections?
Not all the time.
Oh, okay.
Not all the time. Okay, let's say use of contaminated instrument. Okay, and you can get that. So now for the coronavirus, so many people are sick, and I don't know how much precaution they are taking. Okay, so for all those instruments, you can get that Clostridium perfringens. They actually make spores. You can get that Clostridium infection. Another example of iatrogenic, you can say the after radiation, after chemotherapy, what are the things you get? Chemotherapy, like vomiting, nausea, anemia. You can get anemia for radiation. After anemia radiation, you can lose your hair lost, all those things. So these are actually the example of iatrogenic. So these are the iatrogenic disease if it occurs because of a medical procedure, like that vomiting. It's not the pathological infection. It's not from the pathogen. That vomiting you are getting because it is the reaction of that radiation or chemotherapy. You are losing your hair. It's not from the vitamin B complex. Okay, it's because you are getting you had radiation. So a side effect of drugs, complications from the surgery, all these occurred because of that surgery thing. Okay, so these are example of iatrogenic disease. Now, nosocomial disease, these are disease that occurred in the hospital settings. Okay, means you are in the hospital, and that's why you are getting, like, lots of people nowadays get that MRSA. You can say MRSA, VARSA, from the nurses' nose. They carry that Staphylococcus aureus. And we are going to get that nurse she is the carrier, and from her the patient can get. And it can only happen that if that patient is inside the hospital. Okay, nasogenial, you can say like you can get this one also, like improper use of contaminated instrument. That also could be one type of nosocomial disease, that like nurse checked your fever, and thermometer, she forgot to change the cap of the thermometer. Okay, and thermometer. So when she did that, and you can get the same disease from the previous patients. Lots of people who are admitted inside the hospital, they have a very low immunity. Okay, they suffer from poor immunity. So if the person with, let's say, pneumonia is there, any 8 patients, she or he easily can get that pneumonia from that hospital. So these are called the nosocomial disease. Zoonotic disease with the animal vector means they actually, all these animals, they actually carry the pathogen, and they are called zoonotic. Zoo means animal, so they are carried by the animal vector, so that's why it is zoonotic. Like malaria, if you get bite from Anopheles mosquito, then only you are going to get. Rocky Mountain spotted fever, if you get the tick bite, then only you are going to get that disease. So in these 2 cases, that mosquito, so mosquitoes and ticks, they are the vector, means they carry the disease from one person to another person, so that's why they are the vector. Okay, and these germs or these pathogens are carried by the animals, so that's why these are the zoonotic disease. Non-communicable disease like tetanus, legionnaires, like they are not spreadable from person to person. Like tetanus, to get that disease, you have to have bacteria, that Clostridium tetani. So if now that Clostridium tetani, they are the soil bacteria, okay, and they make spore. So if you have any cut or any wound, open cut or open wound, and that Clostridium tetani spore can enter inside your body through this cut or a wound, then only you are going to get that disease. Otherwise, you are not. And anybody who has tetanus, you are not going to get that disease from that person. That Clostridium tetani, they have a special route. It has to enter through that cut and wound, and it has to mix up with your blood circulatory system. Then only you are going to get. You are not going to get it from somebody else.
Dr. Nandi, I have a question. Between the 2 first diseases, I mean, the type of transmissions, which is like a big difference, because they look very alike.
Which one?
The 2 first ones, the communicable and the contagious. They're all being spread from person to person, direct or indirect.
Yeah, that's what I told, that not all contagious disease are communicable. Sorry, not all communicable disease are contagious disease. Because communicable disease, like gonorrhea, it's a communicable disease, but it's not contagious.
Okay.
Okay, so to get the gonorrhea, you have to have intimate contact with that person. Versus the measles, flu, all these are very contagious, like coronavirus, now COVID-19, very, very contagious. And so basically, who are contagious, they are both contagious and communicable. But like as I told, all the sexually transmitted disease, they are communicable disease. They can spread from one person to another person, but they are not contagious. Get it?
Yes, ma'am. Thank you.
So types of non-infectious disease like inherited. Okay, it's genetic disorder like sickle cell anemia, as I told, that Huntington disease. All those are you are actually you have to inherit from your parents. Congenital disease that is present at or before birth, means during the birth. If, like, let's say, basically in this case the congenital, like the embryo, when the fetus actually is inside the mother wombs, okay, it can get that disease, like let's say congenital heart disease. Let's say that congenital heart disease that fetus can get inside the womb, mother womb. How? Because if the mother does not care about the child, like during the pregnancy, mother is drinking, okay, during pregnancy, drinking, maybe taking drug. In that case, that baby can get that congenital heart disease, and it happens during that embryonic development inside the womb. So it started before birth, and during the time of birth the baby is carrying, and its effect might last forever. Okay, so that's the congenital disease. Degenerative disease, progressive irreversible loss of function. Parkinson disease affecting central nervous system. Okay, person lost control of its nervous system. In that case, it is progressive. Very quickly it goes towards the worst scenario, and it's irreversible. Once it starts, we really cannot treat it. Nutritional deficiency, like lack of any type of nutrients, like scurvy, vitamin C. Endocrine disease, like disease involving malfunction of glands that release hormones to regulate body functions. If you have hypothyroidism, like thyroid does not produce enough thyroxine hormone, and that thyroxine hormone, the T3, T4, they are very, very important for your metabolism. Okay, like insulin, if pancreas does not secrete that insulin hormone, in that case the person is going to get that diabetes too. So that's the endocrine disease. Neoplastic abnormal growth means benign or malignant. It could be benign or malignant, like any type of cancer, like brain tumor and all those. Idiopathic disease for which the cause is unknown. We really do not know the the reason behind the disease, like idiopathic juxtafoveal or retinal telangiectasia. In this case, the dilated twisted blood vessels in the retina of the eye. So basically, red eyes. So make sure to study all the definitions. Now, periods of disease, how actually the diseases move or progress through your body system or inside your body. What are the different stages are there? Okay, so there are 5 different stages are there, like incubation period, prodromal period, period of illness, period of decline, period of convalescence. So incubation period, you can see the number of particles is red, and severity and signs are blue. So that blue cycles is the signs and severity, and this is the number of pathogen. Okay, so incubation period, you can see that a number of pathogen, it's slowly increasing the number. Okay, very slowly. But the signs, symptoms, steady state. Really, during that time, the patient does not have any signs and symptoms. He doesn't even know that he's he's having that disease. Sorry, just a second. Okay. Now the next stage, the prodromal period. Okay, it's increasing. It's not straight. It's not going like very steady state, but it's increasing. This part. Well, we start seeing the signs and symptom at a very straight, rapid. Then it slow down, and then again rapid. So the patient is experiencing some signs, symptoms. Sometimes it's very severe, it slows down, and it severe. Period of illness, okay, both are very straight, very severe. All the signs and symptoms of diseases are there. Period of decline, and between period of illness and period of decline, there is a steady state of signs and symptoms. Like once that signs and symptoms, it goes to the peak, okay, and then after certain period of time, it's static. It remains same for few days. Like for the coronavirus, that pneumonia, that pneumonia lasts for 2 weeks or more than 2 weeks, so minimum 2 weeks. So that's the 2 weeks period. Then period of decline, and after that period, slowly the signs and symptoms goes away, and the number of viruses actually goes down. A period of convalescence. Most of the diseases during that time, there is no signs and symptoms, and number slowly decreases. Now, what are the reason of this? What happens inside your body? So the first one, incubation period. Okay, we know that no signs and symptoms of disease. Why? As pathogens just started multiplying, and hence insufficient number of pathogen inside the host's body. For each disease, for all the disease, there is a specific number of pathogen. We call that incubation dose. Okay, that incubation dose actually varies from pathogen to pathogen. Like let's say it's tuberculosis. Now, for tuberculosis, you just need maybe 30 bacteria. If 30 bacteria goes inside of your body, you are going to get, because that your body most of the time is not capable of fighting against that bacteria because they are strong. There are few diseases like you have inoculation dose, like let's say 1 million. In that case, what needs to do, that 1 million bacteria, if all 1 million bacteria inside, then it can create some signs and symptoms. So that incubation dose actually varies. Okay, during this time, that incubation period, at the beginning, let's say the coronavirus, maybe 100 virus we need to have at least the effect. Okay, it could be mild effect, but at least 100. So for a healthy person, that person's immunity, if they can fight, okay, then that person is not going to get any signs, symptoms, and any infection, because that immune system is not allowing that virus to multiply inside or to make colony. Okay, so in that case, we will not have any disease. But for unhealthy person, if virus can enter inside cell and they can start multiplying then slowly. So 100 virus versus when they reached and let's say 210 billion virus. Okay, so that has signs, that creates signs and symptoms. Okay, so it takes time. So to go from 100 to this stages, it takes time, and it, you know, like this time period is called incubation period. Few people get signs and symptoms at the beginning, few people get signs and symptoms here, few people get signs and symptoms here. It's based on that person's immune system. Okay, so that's the incubation period. Incubation periods can vary from a day or two. In acute diseases like for flu, okay, it's you are going to get within 1-2 days. That sneezing, coughing, it starts within 24 hours actually. There are diseases that takes months to years in chronic diseases. Okay, like shingles. So for long time, that herpes virus, it takes long time. That's the incubation period. Length of incubation period depends on the strength of the pathogen, how strong the pathogen. You can, if you compare the coronavirus versus let's say the just regular flu, okay, or you can if it's swine flu. So all these three are same category. They all belongs to the corona virus, that category. Okay, but if you compare the strength, they all varies from each other. Strength of the host immune system depends. Like now you are frequently hearing that, okay, so carrier means their immune system are very strong. They are healthy person. Their body is fighting. So that's why they are not going to get the disease. They are not, their immune system is not allowing that virus to make colony inside their body. Okay, so it depends on that. Site of infection. Okay, so for in case of corona, it's the lungs. So it's out of our control. If it is a skin, okay, so if it is skin, like just a wounds or anything, so we could have get the medicine within 1 month. The people can make it on within 1 month. If it is brain, it could take maybe 20 to 50 years. It is lungs, so it will take maybe I would say 2 to 5 years. Okay, and the size infectious dose received, how many bacteria you got. If for coronavirus, let's say that infectious dose is 100, and if you get 10, nothing will happen. All the virus, they will go all the way up to the throat or maybe inside the nose, and they will die. Because it's very small, but dose, small amount, your body can fight within few seconds or few minutes. So they are not going to create an infection inside this period. Prodromal period, pathogen continues to multiply.
Dr. Nandi, do we know how long people are considered carriers for the coronavirus?
15 days.
15
2 weeks, yeah.
Okay.
Within 2 weeks, they start showing all the signs and symptoms.
So what about someone who isn't going to show signs and symptoms because their immune system?
That means they are healthy. Their immune system is fighting.
So they still are carriers for like 15 days?
Yeah.
Okay.
But in that case, it varies. That 15 days could be short. But usually for moderate immunity, they can survive for 15 days. Usually like let's say within 15 days, it takes for the virus 15 days to express the signs and symptoms. That's the incubation period. During that period of time, that 15 days period of time, people will not show any signs and symptoms because it's a new environment for the virus and it takes time for the adjustment and to fight against the person's immunity. Okay, if it is carrier, what happen, they might start showing very little bit of signs and symptoms maybe around 10-12 days, but most of the time, if you know, it's based on their age, based on their immune system. Okay, for the carrier. So prodromal period, pathogen continues to multiply and the host begins to experience general signs and symptoms of illness, which typically result from the activation of the immune system. So after 15 days, that person is going to show the signs and symptoms. So during this period of time, during that 15 days, people didn't even know that he already got that virus or already got that pathogen. Okay, now from here that person is going to get that signs and symptoms. Now one reason for that signs and symptoms is his body, his immune system started working. Now what can happen? Let's say for the coronavirus, we must have minimum amount, maybe let's say 1,000 coronavirus has to go inside. We still do not know what's the dose for coronavirus. It's too new to tell, but let's say that it's 1,000. Okay, and if you get just only 30, so it's too little, and you know, these bacteria virus, they are not going to survive inside the body, and your body will kill all those organisms. Now they are so too small in dose that your body is fighting, but while fighting, body does not have that immune system does not create any chemicals, it does not secrete any chemicals, or it does not create any situation that may create the signs and symptoms. Okay, like I can give you an example so you will get it. Like prodromal period, lots of people start, not lots of people are usually going to start getting signs and symptoms. Most of the time that comes from the immune system. Basically, when there is a foreign body inside your body, okay, foreign protein, like it could be any pathogen, your body start fighting your immune system. Okay, in that case, body's first response, body start secreting pyrogen, one chemicals. That pyrogen increase your body temperature, and that's how you get fever. Okay, so fever in this case, this is not the this fever, this temperature is not coming from that virus or bacteria, not from that pathogen. It's your body's immune response. Okay, so what I wanted to say over here, if it is only 30 pathogen and the dose is 1,000, it's so minimum that body is still secreting pyrogen, but that pyrogen, the amount of pyrogen is not that much, or is not enough to increase your temperature to a visible level or to a measurable level, so you can consider it as fever. Sometimes you feel like, okay, feverish. It's not fever, I don't have temperature, but somehow I feel like I have fever. What does it mean? That body is making pyrogen, you know, but it's not that level, so that you know, body temperature can increase at least 1 degree centigrade. It's not measurable, but still body is making pyrogen. So not all cells has increased temperature, but few of the cells, body cells, got that temperature. Okay, so that's the prodromal period. You start showing that signs and symptoms. Like for the flu, first 1-2 days, you start, sneezing, coughing, and all the things. Maybe the first day, that is because of the immune response. The second day, yes, you are going to go to this period, illness. So next stage, illness, signs and symptoms of disease are most, obvious and severe. Period of decline, number of pathogen begins to decrease because each cell has capacity. Okay, each cell has capacity, that how many pathogen or how many bacteria it can carry. So each cell reaches to that capacity, to that level. Okay, so cells cannot carry anymore. It cannot multiply that virus genes because the cell is already going to die. It reaches to that capacity. So signs and symptoms of illness begin to decline because number does not change that much. However, patients may become susceptible to developing secondary infection. This is the time, body. So during this period of illness, this is the time like body is fighting at its extreme level. During this period of time, period of illness, okay, period of decline, during this time, body is exhausted. Body cannot fight anymore. Okay, so that's the chance that body is going to get secondary infection, and that's what is happening for the coronavirus. At the beginning, it starts with the dry cough, fever. It's exactly same sign and symptoms like flu. Okay, but this is the time, the period of decline. So 15 days person does not know. During the prodromal period, at this time a person start having fever. Okay, this time high fever, really high fever. Okay, and then starts that dry cough, very severe. Okay, during this time that person get pneumonia. Okay, and if it does not have that support from the ventilator and like breathing machine, then the person is not going to survive. So immune system cannot fight anymore. Period of convalescence, that as I told, the cell reaches to its certain limit. Patients generally return to normal functions, although some disease may inflict permanent damage that the body cannot fully repair. There are side effects and the damages that came from all these periods, so all those things are there. Like for measles, the person, there is no signs and symptom, patient is generally returning to its normal function, but still all the dry rashes, the rough dry skins, all those can stay. So those are there, but infections are not there anymore. So that's the period of convalescence. Acute infection, an infection characterized by sudden onset, rapid progression, and often with severe symptoms but short-lived, like flu, influenza virus, flu virus. Within 7 days, everything is done. Not even 7 for flu virus. Within 5 days, everything is done. Okay, that incubation period takes, just one days. From the second day onwards, that signs and symptoms start. 3-4 day, these 2 days very severe, almost from second to fourth, these 3 days serious, and then 5th days it's the convalescent. They are leaving the body. From 6th day onwards, patient is completely cured actually. Chronic infection, pathological changes can occur over longer time spans, like months, years, like gastritis, that Helicobacter pylori, that I think Bacter pylori. pylori. So for these bacteria, they have that urease enzyme, okay? And what they do, if it is your stomach gut wall, outside of that wall, we have that mucus layer, okay? So what it does, by secreting mucinase or urease enzyme, it actually can dissolve the mucous membrane. If it can dissolve the mucous membrane, is nothing is, let's say this area, okay? So they can make hole over there. So if they can dissolve the mucous membrane, they can make a hole through the gut, and that's what happened, then gastritis. So you have ulcer. In that case, it is called ulcer. You are going to have bleeding and all those. Latent disease, the pathogen goes dormant for extended periods of time, like varicella zoster virus for the chicken pox, herpes simplex virus, all are latent disease. So how pathogens cause disease? You have studied, I think first or second chapter, that Koch postulate. We still actually follow that Koch postulate. So how to identify the disease? How are you going to diagnose the disease? Okay, these are the ways to diagnose the disease. So the suspected pathogen must be found in every case of disease and not be found in healthy individuals. So for coronavirus, we really do not have test kit at the beginning. At least we really did not have anything. But how we could identify? Because by seeing all the signs and symptoms, okay. And when we tested the blood, so for all the cases, that blood was carrying, for all the patients, blood was carrying the same antigen, same protein. Okay, so that pathogen must be found in every case of disease and not be found in healthy individuals. One healthy individual should not carry that antigens or should not carry that pathogen. The suspected pathogen can be isolated and grown in pure culture. So if we can get the blood from one infected person, and we can make a pure culture, we can grow it in lab. A healthy test subject infected with the suspected pathogen must develop the same signs and symptoms of disease as seen in the postulate 1. So same signs, symptoms, or whatever the pathogen we saw over here. In this case, we are going to see the same pathogen and same signs and symptoms. The pathogen must be re-isolated from the new host. In this case, what we can do, so from this person, from the second person, we can isolate or re-isolate the pathogen, and we can introduce that pathogen inside another patient, and exactly same scenario we can see. And in that case, we can grow that pathogen from that blood. So these are the ways that Koch told. We still do that. Okay, if you go to a doctor with flu, doctor is going to withdraw your blood. They are going to take, and from there they culture, and from there he or she start treating you or identifying the bacteria. So limitations of Koch postulate. So that Koch postulate we still follow, okay, but now there is a little difference, okay, because of the invention of technology, because the development of science, okay, so or improvement of the science. So postulate 1. What it told, that suspected pathogen must be found in every case of disease and not be found in healthy individuals. But now we know, like let's say E. coli, the person, it's called AHEC E. coli, okay, it gives us severe diarrhea, okay, but we all carry that E. coli, right, because it's inside the gut. It's a coliform bacteria. Okay, so it's not all healthy individual they carry E. coli, but this postulate is not true. The suspected pathogen can be isolated and grown in pure culture. Now we know that we cannot do that. The virus, We cannot make a pure culture because they are obligate intracellular pathogen. They must have a living cell, so we cannot grow that. Healthy test subject infected with the suspected pathogen must develop the same signs and symptom of disease as seen in the postulate 1. That's also not true because it depends on the health condition of that patient, or you can say the immunity of that patient. Like we cannot compare a patient, HIV patient and a regular healthy patient. We cannot compare that person, these two persons. Okay, so it not necessarily it can happen. We can get blood and we can inject both of these person, but both of them are not going to get the same reactions. Like for coronavirus, there are carrier who does not show any signs and symptoms, or maybe very mild signs and symptoms, and there are patients who are severely sick. That depends on the immune system. So these are actually the limitations of coach postulate. So coach, for a long time ago, whatever he described, whatever he experienced, now with the development of the science, we actually changed a little bit. So the phenotype should be associated only with the pathogenic strains of a species. Like I gave you that E. coli. They are the normal microbiota of our digestive tract. Okay. So when we are looking for the E. coli, we are not actually looking for the E. coli. We are going to look for a specific strain. Okay, is it a normal E. coli or the EHEC version of the E. coli? We have EHEC, we have ATEC, so many different types of E. coli are there, so many different strains of E. coli are there. So that postulate is correct, coach postulate, number 1 postulate is correct, but instead of seeing the pathogen, if we change the version a little bit, the vibrage, like instead of pathogen, we have to tell that it is pathogenic strain. Okay, so one specific strain should be there, not that pathogen. Inactivation of the suspected gene associated with the pathogenicity should result in a measurable loss of pathogenicity. What we can do, that simple E. coli, okay, we can convert this E. coli EHEC, or we can convert the this E. coli into ETEC, or oppositely, we can convert this ETEC E. coli into normal regular E. coli, or EHEC E. coli into normal regular E. coli. How? Just to make the gene, okay, the gene which is making a specific protein, and because of presence of that protein, that version of E. coli is going to be very, very pathogenic. So what we can do, if we can inactivate that gene, so that same bacteria is not going to make the protein, and it will not give us that same disease. In that case, it could be that normal regular E. coli. So nowadays we have all these machinery, and we can do that. So all these things we have to taking into account. So pathogenicity and the virulence. How much time do we have? Okay, so pathogenicity, the ability of a microbe to cause the disease. Okay, and it depends. Okay, it depends on so many different factors, and that is the degree of pathogenicity in a microorganism. Okay, like coronavirus, few people they have a mild, few people they have very severe kind of disease. Okay, so that degree, that scale from that both the ends of a scale, that's the virulence factor. Many properties that determine a microbe's pathogenicity or virulence are unclear or unknown. We really do not know. But when a microbe overpowers the host defense, infectious disease results. Okay, we know at least this thing, that if a microbe is more powerful than the host defense, then we are going to get the disease. So best way not to get the disease is to improve your immunity. Okay, that's your defense. We measure that virulence factor in two different ways: ID15 median infectious dose and LD50 means median lethal dose. So ID50 median infectious dose, the number of pathogenic cells or variants required to cause active infection in 50% of inoculated animals, means test population. So what does it mean? Let's say we are applying, there are 100 test population, 100 men are there. Okay, we are testing that coronavirus onto them. Okay, and all these person are going to get that coronavirus antigen at same percentage. Okay, now let's say this group, they are going to get 10% of antigen. Another 100 people, they are going to get antigen, maybe in this case 20%. Another 100 people, they are going to get antigen 30%. Okay, now we have 3 sets of test population. They are going to get the same antigen, same pathogen, but the dosage are different. Okay, and our test population, the number actually same. Now in this 3 scenario, which dose is going to kill 50% of the test population? That is our infectious dose. Okay, so over here our infectious dose was 30, over here our infectious dose was 20, over here 10. So 10% antigen means we introduced 10 virus, we introduced 20 virus, over here we introduced 30 virus. So each person got 30 virus. Okay, each person got 20 virus, like that. Now let's say over here this is the dose, like this set of test people, they got 20 virus, each of them got 20 virus, and in this case after the testing, if 50 people get that active infection, so out of 100, so we introduced 20 viruses to 100 people, and after the test, if we see that 50 people are sick, that is the ID50 median infectious dose. Okay, because this percentage, this is the most active percentage that can kill 50%, not kill, that can give the infection on 50% of the people. Okay, that's the ID50. Exactly same way, if it can kill 50% of the people, that's the LD50, means that dose is actually lethal on those test population, and it can kill 50% of that test population. So ID50, the difference between these two, it can kill. Over here it can infect, means they can make colony inside, they can make infection for 50% of the population. You can see over here ID15 doses like norovirus. This is for hepatitis. Hepatitis ID50 is 10 to 100. ID50 means if it is hepatitis B, hepatitis A, so only 10 to 100 virus should be enough to make 50% of the people. Okay, parts, as you can see, which one? Let me see over here. Giardia lamblia, that's the protozoa, diarrhea. Just one protozoa is there, then 50% people can be sick by just getting one protozoa. So that's the so all organisms, all pathogens, they have separate dosage. So how are we going to find out? So this is the number of pathogenic agents, number of bacteria, and this is the percent of mortality. Like this is for LD50. it could be ID 50. So this could be the percent of infectious person. Okay, so that's the number. So what we have to do, we have to see that where this side and this side, so we have to draw a straight line over here, and we have to draw a straight line over here, because it is, we are going by 50%. Okay, so this is the line, and then first draw that line and then pull it down, so 10 to the power 4. That's the number for this bacteria for the LD50. So this number, this mini virus is lethal for at least 50% of the test participants. So molecular determinants of pathogenicity. So we don't have time. Okay, so let's keep over here. I will start then. What?
Miss Nandi,
Mm-hmm.
have you put the sample questions up yet?
No, I didn't. I really did not have time this weekend, but I'm going to do
Okay.
as soon as possible. Yeah, I'm going to.
Okay. Okay.
Is
Thank you.
is this next test going to be like the previous one?
Yeah, mostly.
Okay. So like practice the sample.
But there will be few chapters that you have to study by yourself. I hope you know that.
Oh, because I know that they weren't in the updated syllabus.
No. There are few chapters that you have to study. I
Okay.
will tell you, and for that I'm going to give you some question. If you study only those questions, then you should be fine.
Okay. Yes, ma'am. Thank you.
Okay, so I will see you on Wednesday.
Yes. Thank you.
Okay, good night.
Good
And
night.
be safe.
Miss Nandi, are you gonna do are we gonna have to do the infographics and the discussion for the last test?
Oh, yeah, I completely forgot about that. Yeah,
Miss Nandi,
post.
it's like, I wonder if you've always done that at the end of every semester, because there's no way you can cheat with the Respondos lock thing.
Mhm.
So our tests are not easy just because we take them at home.
Mhm.
So would you consider not making us do the infographic and the discussion post for test 4? It is a lot of work, Miss Nandi, and all teachers are giving us more work because they think that because we're at home, we have less things to do. And you being at home know that that is not the case.
I know, but that's the instruction we got. Like we are following all the thing university system of Georgia wants us to do. So there are few things I really.
Miss Nandi, when you're in a class that is like online based, because I have taken a bunch of those, you don't actually have to sit in a classroom for hours, which we have to do with your class and every other class because we used to be in-person students. So we still have to attend classes, and also we have a bunch of more homework, like if we were online based students.
I understand.
It's very time consuming, time demanding, and we also need to study for your test because that's the point where we have like the most points to acquire. That's like the most important thing of the class. And when we're wasting time doing infographics and
if you all are ready to study by yourself, I can just post the PowerPoints are there. I can just post the sample question. In that way, you don't have to, then we can cancel the rest of the Zoom session. It's up to you.
No, because the Zoom sections are even better than doing infographics and doing discussions. Like it would be better to cancel that out because we weren't even counting with it at the beginning of it.
Exactly,
Just have
because
bonus.
Instead of no, I have to give that.
Because I mean, if you just count our tests like they were supposed to be 100 a piece, we'll still have the test that we just took. We have a fourth test. I'm sorry, and we also have the comprehensive test.
Okay, this time I'm going to give you easy infographics and easy discussion. I promise, and I'm going to give you two for both. Okay?
And wouldn't be able to maybe just have one instead of two? It's just a
No,
lot.
we have to go by two, but I will make it short. I got your problem. I will
just
make it short and I will make it easy. Okay?
right now we are confused. We don't even know where to go to see the questions. Everybody's like confused. And
Yeah.
I mean, we wouldn't even think about not doing the lecture because the lecture is valuable. Miss Nandy, you give a lot of relevant information that we need to have. I wouldn't even think of canceling the lectures, but please talk to someone so that they don't make us do the infographics and discussions. It's a lot of
No,
work.
but for infographic and discussion, I posted note on D12 that okay. So I posted video for infographics and I posted that discussion question. I gave you direction.
But we still have to do some research and study to do a good
Yeah,
job on it.
that you have to do. But at the same time, if you read the text, look, all those infographics and discussion, that question came from chapter 9 to 13.
9
That
to 13. Okay.
yeah.
Thank you.
So that's what it came from. So you are supposed to study all those chapters and you know you should know the answer. So but I gave you direction where to find out the question.
Okay, I do have one question. Depending on how we did on just the test questions, if and we got all 70 questions right, if we didn't do the infographics and the discussion, it would just be 70 out of
Class.
the points, right?
That's the 70%. Yeah, you are going to lose that 30%.
Yeah.
Okay.
Yeah, but you know what? Why I gave you these two? Because these two are easy to grab points.
It's not
Infographic,
easier.
no,
It's
but
not easier because
We already studied a lot for the test.
we're studying a lot for the test, and we're also having to study to do something else that we weren't going to
but
study.
if you have studied for the test, then you already should know the answer for those discussion and the infographics. Because look, for the test you had to study from chapter 9 to 13, right? And I gave those topics from chapter 9 to 13. So if you have studied for the test, then you already know that answer. You should know the answer. Okay, so now I promise, you know, for next discussion, I cannot change it now. I have to go through that, but this time I will make it easy, okay? And I'm going to give you clue so you know which chapter.
And because what we're fearing is, okay, we studied a lot, Dr. Nandi, a lot for the test, and you can see my grade, I did well. But I'm afraid that I'm not going to be able to do the discussion post and the infographics up to standards, and I'm going to lose points there when I already did my best on the test.
Okay. So this time I'm going to make it easier. I promise. You really do not have to work that much, okay? And for infographics, let's see what I can do. Let give me time and let me think.
Okay.
What I can do.
We would really appreciate if we didn't have to do that.
No, that I cannot do, Esmeralda, because we have to. Because my syllabus already is there. Not on digital, I'm talking about in the division in the college. So it's there, and if they find out that now I am changing, then I will be in trouble. So now I cannot do that. I cannot take out that.
Who do we have to talk to? Because I feel like they're just not taking into account what's going on. Like they're just acting like this is just school that we're doing at home, and that's not true. Like this is an unprecedented thing, and we're at home with our kids and we're trying to study with our children and, you know, go to work weird hours now.
They're just
and
dumping more work on us. Who do we talk to? Yeah, Autumn is right. Who do we have to talk to?
Okay.
not normal time.
So how about I will give that 10 points. Okay, infographics you don't have to do. I'm going to give all of you that 10 points. But discussion you have to do.
Thank you.
Thank you.
Thank you.
In that case, I am going to let me see how easy I can make.
Thank you, Ms. Mandy.
Infographics or
So
just
what if we try the infographic and we,
No, she already said something.
okay.
Let's be grateful.
Yeah.
Let's be grateful.
I could study all day for a
that's-
test. I
it's
could
great.
study all day for the test and I never do good.
But
So
what I think that infographic helps for the people that need those extra points. But what about the people that can easily just ace a test?
No,
and we don't need the extra
no,
point.
that's the thing. Look, the infographics tell me about the critical thinking.
Okay,
Yeah.
she already said that she's
That's
going
why
to give us a 10
if you
points.
understand, look, that most of the people, what they do, they just memorize the sample question. Okay, you should not do that. You have
That's right.
to understand the concept. If you understand the concept, then you can work on the discussion and the infographics.
Yep.
It's getting hard means you didn't get the concept.
That's right. Because I
Like
can
the-
study all day for the test, for a test, and I'm not a good test taker. And I'm working, and I work long hours, longer than usual, except on school nights. And so when I come home, I'm tired, Miss
I
Nandy.
know, I understand.
So
completely feel-
I understand the infograph, but I don't understand what you're wanting. I think I know where the answer is, but I don't quite understand. If I do a PowerPoint, will that be enough?
Yeah, PowerPoint should be enough. Just make one
Okay.
slide. Just one
Okay.
slide.
Okay.
Try to add three pictures.
Okay.
And 5 facts.
Okay.
What do you think? What could be the possible explanation? Okay.
'Cause-
Like let's say on one plate, if we see 3 different colors of the colony, that means you can conclude that 3 different bacteria are there.
Yeah.
Okay? So that's one. By looking at the colony color, you can identify that how many different type of bacteria are there. By looking at the colony shapes and sizes, you can clear. So over here, the shape 1 point, color 1 point, size 1 point. So exactly like that way, I want 5 facts. Just what are you thinking that it could be?
Okay. Because I see where everybody's coming from. I understand. I get what they're saying. Some people are good test takers, and that's awesome. I'm glad for them. They're great. I wish I could do that. I retain what I use, what I remember, what I work with on a daily basis. I looked at the test afterwards, and it's very well everything that I do for the last 17 years I retain. I can't retain all this other.
Well, Vivian, it is not that I am a great taker.
Yeah.
It is not that. I took 2 days off to study for this test.
See, and
I'm not
can't.
getting paid for those. I'm not getting paid for those, but I think this class is worth it because I need to be admitted. So it is a lot of work. I mean,
I had a day off from my work too, and I almost got a warning for it, and I had to do it because I wanted a great grade.
Yeah.
So yes, I work myself off for this grade.
So that test costs me a lot of money, Vivian.
Yeah.
Because I had to take the day off. So Miss Nandi had already said that she was going to give us the 10 points. And you kept delving on it.
I'm going to give, not for discussion, for the infographics I'm going to give. But for the discussion.
Okay, we'll do the discussion, Miss Nandi. Thank you very much.
And I will make it easier, don't worry. Okay?
Okay, thank you very much. Sorry to take your time for this.
Okay, good night.
Good
I will
night.
see you on Wednesday.